Pleasure to introduce today Thomas, or as those of us who know him well, Trey Westbrook. Trey comes from a little bit from everywhere across the country. He was born in Mississippi, spent some time in Hawaii, and did most of his training up in the Northeast in Allegheny. I think you were an academic All-American, not so bad at tennis, I think, back in the day. Did his graduate studies in Rochester, and also finally did his postdoctoral training with Steve Elledge at Harvard, and I think that's where he made a number of important contributions, notably in collaboration with Greg Hannon and Scott Lowe, developing some of the really breakthrough or innovative tools that most of the field uses to perform functional genomics in humans. Trey has been a tenured associate professor at Baylor College of Medicine, and he joined the faculty at Baylor in 2007. Since that time, he's also been director of their functional genomics core. And I've had the pleasure of knowing Trey for a very long time. He's one of the most insightful people I know, at least when it comes to science. Very infuriating because he's, in our collaborations, he's always the one to push us to ask the next question, to dig deeper. And I think that his work really exemplifies how, when applied properly, functional genomics can be a very powerful engine for discovering translationally relevant findings of great impact. And that's what he's going to share with us today. Thank you, Trey.
Thank you, Don, for that overly kind introduction, and also for the invitation to be here with you today and for the opportunity to meet with colleagues here at Yale. Certainly this is a place of great tradition in innovation, both in science and in medicine, so it's a real privilege to be with you here today. As Don mentioned, we spent really the better part of a decade now developing new genetic technologies for forward, or sometimes called functional, genetic screens in human and in mouse systems. And much of my lab spends its energy trying to leverage or capitalize on those technologies to answer fundamental questions in cancer, both from a basic science and also from a translational perspective. And so today what I decided to do is to share just a couple of vignettes. I won't talk too much about the technology, but happy to do so if there are questions in the Q&A. But really wanted to just share with you a couple of stories of biology where I think the tools are just giving us new entry points or new insights into how we might attack problems in the clinic. So the studies I'm going to describe to you today are largely based in the context of a subtype of breast cancer called triple-negative breast cancer, or TNBC. But I believe the gene networks, the principles that we've learned probably have a much broader impact even beyond this important disease. So I recognize there's many breast cancer aficionados here in the audience, so I'm not going to belabor much of an introduction. Please let me just remind you that this subtype of breast cancer accounts for nearly 250,000 new diagnoses each year. So even if it was considered its own disease, it would still be the 5th most common cancer in women. And it commonly progresses to metastatic sites of the lung, bone, brain, and liver. And that's a point I'll come back to in just a minute. So in contrast to the two other major clinical subtypes of breast cancer, that would be ER positive and HER2 amplified, this disease really has lacked, therapeutic traction. And that's been for a number of different reasons. One can say we simply don't understand the genetic determinants of this disease. What are those pathways and networks that seem to drive the pathology? And I would say a step beyond, we really don't understand what are its vulnerabilities. As a geneticist, it's my bias that every genetic perturbation comes with a cost. And so I believe that every tumor has its own set of vulnerabilities, and the question is how we try to elucidate those changes and those weaknesses. So the stories I'm going to tell you about today really arise from two simple challenges, again in the context of TNBC. One, what are the genetic underpinnings of this disease? What are those gene networks that actually play a role? I think we've got some fantastic and fascinating characterizations of TNBC genomes, epigenomes, and now proteomes, but we still lack a fundamental understanding of what are the gene networks that are underlying this disease. But the first vignette I'm going to share with you is really how we've been discovering new types of stresses or addictions, if you will, in triple-negative breast cancer. And then some of our efforts, our early efforts to translate those basic science discoveries towards the clinic. Okay, so as you well know, common oncogenic drivers, your Myc and RASs of the world, they certainly confer many of the pro-tumorigenic hallmarks of cancer, and much of the cancer community, for good reason, focuses on understanding how these oncogenic perturbations give rise to these cancer behaviors, if you will, with the hopes of essentially intervening in these various behaviors and trying to revert those effects. But our group has been largely focused on the alternative side of this balance, trying to understand how is it that drivers of breast cancer can confer new or heightened stresses onto cancer cells and the surrounding environment. Now, the literature is certainly rich with examples of how oncogenic lesions can confer different types of stresses, from evoking apoptosis, senescence, metabolic rewiring, and even changes into the host stroma and tumor or immune microenvironment. But it's my opinion that we're still really just scratching the surface of the kinds of stress phenotypes and the kinds of addictions that are caused by these oncogenic lesions. And I think that's primarily been based on our approach. I think that while the genomic revolution has certainly given us fantastic insight into what the disease looks like, in its nature it is by definition correlative, and I think that what is needed is really a complementing attack on the functional side. Now, I can say that I think many of you are already in that boat and driving, and certainly there's some thought leaders here. But I would say that that type of functional work has lagged a generation or two behind from the genomics community. And so I would certainly advocate for more of that type of work. So the vignette I'm going to share with you today was really our attempts to understand the underlying stress phenotypes and addictions conferred by the Myc oncogene. Now, certainly Myc is a driver of cancer well known to many of you, so I'm not going to belabor much of an introduction, but we chose to tackle the Myc problem from some simple-minded reasons. I was not a Myc biologist when we got into this problem, but certainly it's hyperactivated in many cancers. The estimates are 30 to 40% of human cancers are driven by Myc. It tends to confer or be associated with the poor prognosis types of malignancy, and so certainly drives a major problem in human disease. Myc is, of course, a transcription factor, and like many transcription factors, this has been an area difficult to gain therapeutic traction because of its many important functions and functional surfaces. And finally, like many classic oncogenes, Myc has this sort of dual nature that I described a moment ago, conferring both pro-tumor phenotypes as well as cellular stresses. And so our goal was essentially to delineate such Myc-driven stresses and their genetic underpinnings by finding the genes that are absolutely required for cells to tolerate Myc. And to get an unbiased view of those type of support pathways, we developed a genetic approach. So here we're using many of the barcoded RNAi libraries that we've developed over the past decade. I'm, again, not going to talk a lot about these technologies, but suffice it to say they enable us to do rapid loss-of-function genetics at scales that prior to the barcode work were really unprecedented. So this type of approach allows us to ask many questions, and we employed the loss-of-function approach to find genes selectively required to tolerate Myc. So the first iteration of this screen has been published, so I'm going to move quite quickly here. Essentially, we've engineered normal mammary epithelial cells with an inducible Myc transgene. This enables us to perform genetics really in two isogenic states. First, we of course can do a genetic screen in the wild type state. This is just simply asking what are those genes that are essential for viability and support of these mammary epithelial cells. But the key to the success of the screen is really the ability to do the screen in the paired or isogenic state. And it enables us to find those shRNAs, like represented here by shRNA-Z, that are selectively required in the Myc-on state, but not in the Myc-off state. And we would define those candidates as genes required to tolerate Myc hyperactivation or Myc synthetic lethal. So from these screens were born out a lot of different signaling pathways and networks that seem to be required to tolerate Myc. And I'm going to tell you about today in just a few short slides about one of the most surprising discoveries that I think emerged from these screens. And that was the identification of the spliceosome, the core spliceosome, as Myc synthetic lethal. Now please let me remind you that the spliceosome is of course a macromolecular machine consisting of several major subcomplexes or SNRPs, and indeed some of the major innovation in splicing biology and function has happened right here at Yale. And this machine consists of over 100 different proteins contributing to its core functions. And as I'm sure you're aware, the prominent function of the spliceosome is of course the splicing of pre-messenger RNA, where it catalyzes several steps in the removal of intronic sequence, shown here in green, and the adjoining of two consecutive exonic sequence, shown here in yellow. And as you can see from this video, this process takes the coordination of several complexes and enzymatic activities, shuttling on and off of the pre-mRNA during its processing. And what was notable from the primary screen is that we actually observed components of each of the major subcomplexes of the spliceosome as required to tolerate Myc. Now shown here is just a representative example from some of the validation. Again, we're using engineered mammary epithelial cells, so still an artificial system, so we can control the sort of Myc normal or Myc hyperactivated state. And now we've engineered them on top of that with inducible shRNAs targeting different components of the spliceosome, asking the question, what is the consequence of interfering with spliceosome function in the normal and Myc hyperactive state? And as one can see here, perturbation of here two components of the spliceosome, SF3B1 and SNRPF, were selectively deleterious to these mammary epithelial cells in the presence of Myc hyperactivation. This is just representative data. We've marched across all of the major SNRPs in the spliceosome and see essentially the same phenomena. And I'd like to make an important aside here. We're not making null alleles when we're using RNAi, so this is in contrast to CRISPR or the like. But instead, with RNAi, we're making hypomorphs or generating partial loss of function of the spliceosome. I think all of us would agree that complete loss of function of the spliceosome is probably deleterious to most of cell viability and to organismal life. But here we're doing much like what a drug does and creating a hypomorphic situation. And indeed, if one actually uses a pharmacologic inhibitor of the spliceosome, you can see a similar scenario where partial loss of function of the spliceosome leads to loss of cell viability in the Myc hyperactive state. There we go. Pardon me for a second. All right. So this and a lot of other data, I think, led us to what is really a surprising observation and really a more confounding question. Why is it that Myc hyperactivation would lead to such a dependency on the core function of the spliceosome? That was something that was puzzling to us, and we certainly spent more than a year scratching our heads about this. But at this point in our study, our model was significantly impacted by a long-standing observation from the Myc community. Now, many of you may know that Myc has long been described to increase rates of protein translation, but what's also striking is that oncogenic Myc enhances or increases cellular transcription across the transcriptome. Now, Myc is, of course, a transcription factor, but I'm not suggesting its role as inducing of tens or hundreds of genes. Recent data has circumscribed that Myc can amplify transcription across those expressed genes in the transcriptome. So in this case, this phenomena of increasing transcription rates has been observed in many different cellular contexts, tissue contexts, and in different types of Myc-driven cancers. And indeed, in our mammary epithelial cell system, one can see that acute hyperactivation of Myc leads to elevated synthesis of pre-mRNA across the transcriptome. And so from that phenomena, I think the model that was provoked was fairly simple. That Myc induces an increase in pre-mRNA synthesis that essentially burdens the spliceosome, which may become a rate-limiting or more rate-limiting process in the maturation of pre-mRNA. So this model provokes a simple set of predictions. One, that spliceosome inhibition would generate significantly more intron-retention in this hyperactivation state. In this kind of bulk model, we're arguing that the enhanced level of substrates, these pre-mRNAs, are overwhelming the spliceosome and causing an untenable situation when the spliceosome is perturbed. So this prediction suggests that in this fourth synthetic state, we should observe significantly more intron retention. Again, not in a few genes, but in a more global phenomenon. And so we asked the question whether or not we did indeed observe that kind of intron retention by doing RNA-seq across these four states. Again, in a normal, Myc-hyperactivated, or spliceosome perturbed in those two cell states. And then measured intron retention using RNA-seq. Now, the ultra-deep RNA-seq here, of course, captures gene expression, but that's not really what we're after. What we're trying to ascribe here is the level of intron retention that is occurring across gene bodies. And so shown here is just an example of one intron, where you can see that loss of the spliceosomal factor BUD31 causes a modest increase in intron retention in these cells. But if the same perturbation, the same level of depletion of the spliceosome factor in the Myc-hyperactivated state significantly enhances intron retention. Now that's one intron. If one takes a more global panoramic view of the transcriptome, you can see the same phenomena. What I'm showing you here is the effect of BUD31 or spliceosome depletion on intron retention across all observed introns in the transcriptome, and that's following this gray line. And what I hope you can observe here is that in the context of Myc hyperactivation, there is a wide shift in that intron retention that is occurring across introns. And the fact that this whole S-curve shifts to the right and not just part of it suggests that this is not really a phenomenon we're studying at the level of a few hundred or even a few thousand introns, but is occurring, again, across this transcriptome. So what does this mean? What's the consequence for this type of global intron retention? Now, as you might imagine, with so many mRNAs harboring intron retention and undergoing quality control, premature degradation, this could lead to defects in many cell-essential genes and processes. And indeed, if we look at the ontology of mRNAs with the most severe intron retention and decay, one sees that many essential cell functions are perturbed. And indeed, if we study those on a more individual level, you can see that perturbation of those processes is, of course, deleterious to cells. Now, what this argues for is a sort of death by 1,000 cuts. In data that I don't have time to show you, we actually think that the mechanism for this synthetic lethality may hone in on another biochemical pathway, and that's something I'd be happy to discuss at the end of the talk. So overall, the data that I've shown you suggests strongly that Myc hyperactivation confers a sensitivity, if you will, to perturbations in the spliceosome. But while the artificial model we've used has certainly helped us to hone in on the genetic interactions, certainly one can fairly ask, what implication does this have in the context of cancer, if any? And I think the obvious prediction is, do Myc-driven malignancies, in this case breast cancers, are they sensitive to perturbations in the human spliceosome? And so, as I showed you just a moment ago, Myc is actually one of the very few well-established drivers of triple-negative breast cancer. And it's been estimated that Myc hyperactivation occurs in roughly 70 to 75% of this disease. Now, to summarize some of our preclinical work, we collaborated with Ben Neal, who was at Toronto and now in New York, who assessed really the effects of spliceosome perturbations across a panel of a little more than 70 immortalized and transformed breast cancer cell lines. And what we observed was quite remarkable, that indeed in those models, Myc-driven or Myc-dependent breast cancer models are much more sensitive to perturbation of the spliceosome. And this is done by RNAi. I should also point out that the synthetic lethality between these two genes is independent of p53, and that's important since triple negative, more than 90% of this disease is mutated or aberrant for p53 function. And I think in data, I won't have time to show you, but it's certainly striking to us, there are other Myc-driven malignancies that also seem to be dependent on the spliceosome function. So we don't think this is something specific to that developmental lineage of the mammary gland or in breast cancer per se. So that's in vitro. What about in vivo? If one does the kind of typical experiments, you can see that genetic inhibition of the spliceosome, in this case with BUT31, certainly confers a phenotype. This is what we would call a treatment experiment, where we've engineered triple-negative breast cancer cells with an inducible shRNA to BUT31, allow them to transplant and engraft, and then after tumor formation, we randomize into with and without induction of the shRNA. And you can see here that even after, I think, about a 3-week exposure to DOX, you get a significant effect on the progression of this disease. But I told you just a moment ago that the spliceosome is really a macromolecular machine. There's more than 130 components to the spliceosome, and so if we were going to systematically ask that same question, that would require quite a mouse budget. And so what we then turn to is our barcoding system, in which enables us to ask multiplexed or highly parallel questions about effects of RNAi of various genes, but in a pooled manner. So the way this system works is we're still engineering triple-negative breast cancer models, Myc-driven triple-negative breast cancer models, with shRNAs targeting individual genes. In this case, I'm showing you a control Myc targeting shRNA, one targeting the spliceosome. In the data I'm about to show you, this is actually a pool of about 300 different shRNAs. And what one can then ask is, what is the effect of inducible RNAi on the progression or the viability of these triple-negative models in engrafted tumors? And what you can see here is perturbation of Myc leads to a significant reduction of these cells in the tumor population, as one would expect from a Myc-driven model. And the same is also true for components of the spliceosome, just shown exemplary here, a core component called FTUD. And so this and many other data suggests that perturbation of the spliceosome is deleterious in triple-negative breast cancer cells. But I told you a moment ago that, of course, rapid metastasis or onset of metastatic progression is certainly almost pathognomonic with triple-negative breast cancer. And so we asked the corollary question, but in the context of the metastatic setting. Here again, we're engineering triple-negative breast cancer cells with inducible shRNAs in a pooled fashion. In this case, I'm showing you control in BUT31. Allow them to engraft into the lung using an experimental model of metastasis. And then upon transplantation and establishment of metastatic lesions, we ask whether inducible shRNAs to BUT31 has any consequence. And as you can see here, partial loss of function of BUT31 certainly deleterious to these lung metastatic lesions. So this was strongly supportive of at least the possibility that perturbation of the spliceosome is absolutely essential for Myc-driven breast cancers and their progression. But I think this translational data is also cheating a little bit, because up to now, all I've shown you is data in which we're perturbing the spliceosome, but selectively in tumor cells. And that, of course, doesn't help us to appreciate any of the possible therapeutic index. And so to do so, we have a now well-established collaboration with the medicinal chemist at SRI, who has several generations of fully synthetic and bioavailable spliceosome inhibitors. Shown here is pseudomycin D6 or SD6. And what I'm showing you here is that even a 2-week administration of SD6 significantly reduced lung metastatic burden in this model of triple-negative breast cancer. So I believe it's still early days. I think that this raises a couple of important and provocative possibilities. One is that the spliceosome might be an area where we can gain therapeutic traction in triple-negative breast cancer. But to put these discoveries in a little bit of context, there's certainly been a lot of activity in the Myc space, trying to revert many of the pro-tumorigenic programs that conferred by Myc. So if one thinks about perhaps many of the efforts from my friend and colleague Jay Bradner or others in the BET inhibitor or bromodomain inhibitor space, those have certainly shown some interesting results in ameliorating some of these pro-tumorigenic effects of Myc. But what we're arguing is that there's a complementary strength in perhaps trying to leverage the stress phenotypes that are conferred by Myc. Instead of trying to revert its transcriptional programs, we're taking advantage of the stress that is imposed by those broad Myc effects on tumor cell transcription. And more broadly, we think that this argues there might be other steps in RNA processing or RNA metabolism that will be interesting entry points to think about intervening in these MYC-driven cancers. And I would like to maybe take a sidestep here and also mention that we don't think MYC-driven breast cancers or that MYC per se is the sole oncogenic driver of these spliceosome dependencies. And indeed, there's been mutations in spliceosome core factors and a number of other transcription factors that we think are good candidates for also conferring this type of dependency on RNA metabolism. Okay, so that's a sort of a view of our work in the RNA metabolism. For the next vignette, I'd like to share with you really a study that began with a more basic science goal of discovering new pathways that govern triple-negative breast cancer. So as this audience is certainly well versed, growth factor signaling plays a prominent role in cancer etiology. And as you know, developing both small molecule inhibitors and antibodies to growth factor receptors has been a major emphasis in the anti-cancer campaigns. And while we can all appreciate that growth factor signaling is regulated at many levels, I think the community's attempts to leverage receptor inhibitors has largely been guided by, of course, somatic mutations in growth factor receptors. So one can think EGFR inhibitors for EGFR mutant lung cancer, HER2 inhibitors for HER2 amplified breast cancer, and the list goes on. So in each of these contexts, the receptor mutation provides both the target for the anti-cancer therapy and also, of course, the biomarker for delineating those patients that might be responsive to an intervention. But in each of these contexts, I would argue that while there's been some varied success for leveraging these types of inhibitors, it ignores sort of the vast majority of cancers that actually do not harbor mutations in growth factor receptors but still have this phenomena of hyperactive growth factor signaling. And I think this provokes an obvious question. Are there alternative strategies or alternative rationale where the armamentarium of kinase inhibitors, receptor inhibitors might be leveraged in those tumors that lack common receptor mutations? And again, we'd return to triple-negative breast cancer because I think this is actually one of the premier examples where it's been long known triple-negative breast cancers harbor hyperactive growth factor signaling in receptors like MET, PDGF receptor, and EGF receptor. But like most cancers, there are really no common receptor mutations that can explain that phenomena of pathogenic signaling. And so for the remainder of my talk, I'm going to present to you, again, a project that began with a basic science goal. Our motivation here was simply to uncover what are the signaling networks that are driving triple-negative breast cancer, and then tell you about how our efforts to understand one of these tumor suppressor networks led us to think a little differently about how we can apply therapeutic agents already in the clinic. Okay. So, as I said, this story really begins with our long-standing efforts to identify new tumor suppressor networks in breast cancer. We're doing this using 4 genetic strategies, so using large-scale RNAi screens. And these screens are based in large part on cell models of malignant transformation that were pioneered over 2 decades ago by Bob Weinberg and many others. Now, many iterations of these screens have been published, so I'm just going to touch upon a few salient details. Essentially here we're engineering normal cells, in this case mammary epithelial cells from a breast reduction. We're engineering them with perturbations in oncogenes and tumor suppressors that even in combination do not transform cells, but instead prime them to transformation from one additional genetic event. And then one can ask the very simple question, does in a gain of function or in a loss of function paradigm, can you tip those cells over the edge and confer malignant transformation? And so we've done this using both gain of function, Orpheome or cDNA libraries, as well as shRNA libraries, and that's the work I'm going to focus on in just a moment. So from these types of screens, we find many of the bona fide, well-loved tumor suppressors that many of us work on, PTEN and the like. But we've also found a number of, about 42 actually, new tumor suppressor candidates, many of which have now been validated in the community by both mouse genetics and in patients. And in some exciting data that I won't have time to show today, we've actually, 2 of these candidates have now emerged as familial cancer syndromes, one of which should be out in publication soon. So the candidate that emerged as one of the strongest hits from these screens was the tyrosine phosphatase PTPN12. And so that's where our focus is going to be here for the last few slides of the talk. So as one would expect from a candidate tumor suppressor from this screen, PTPN12 is itself a potent suppressor of cellular transformation. Shown here, RNAi to PTPN12 is potently transforming in that engineered cell system I mentioned a moment ago. And if one looks at shRNAs to PTPN12, you can see that the dose-dependent effects of its depletion correlate nicely with the degree of the phenotype. Now, I should point out here that even a 50 to 60% reduction of this protein is strongly transforming, and that's consistent with the idea that this might be a haploinsufficient tumor suppressor. The mouse genetics are starting to play that out, and that's a point I'd like to come back to here in just a moment. But suffice it to say, in whatever mouse gem type situation or in human cell models, loss of function of PTPN12 is strongly transforming. So, of course, with a candidate tumor suppressor, next step is what's the evidence of inactivation of the tumor suppressor in the human? And so in pre-TCGA days, we began doing some simple experiments sequencing the locus in a cohort of about 300 primary human breast cancers. And those experiments were both encouraging and discouraging all in one. After sequencing about 300 of these breast cancers, we found a whopping 4 mutations in the PTPN12 locus. That's the disappointing part, all of which were in triple negative. So even if accounting for just in triple negative subtype, that's only 4% of the disease. Certainly a cause for some reservation. But what was nice about the way we discovered it in the cell systems that we had at hand is we could actually ask whether those mutations were indeed loss of function. Now, as I showed you just a moment ago, PTPN12 shRNAs are transforming in these mammary epithelial cell systems. One can suppress that with a wild-type cDNA. But at least a handful of these mutations are clearly loss of function and may actually encode some dominant negative activity. And so that argues that the mutations that we were uncovering were indeed either hypomorphic or loss of function by the ways we're studying here. So if you fast forward to now 2 or 3 years to the pan-TCGA view of PTPN12, we've noted that PTPN12 is actually somatically mutated in about 1% of human cancers. So that's a fair number, and we're in the process of systematically going through those mutations to ask what percentage of those are actually loss of function in the context of both molecular and cellular phenotypes. But returning back to the breast cancer, this is still at a point of, well, is this really the type of candidate one wants to study if it's 1% of breast cancer, 4% of triple negative disease? And so we then asked whether or not there are other mechanisms by which this tumor suppressor is compromised or undergoes loss of function. And so for that, we've developed a number of immunohistochemical assays. And to make a long story short, looking at an independent cohort of human breast cancers, we can see that nearly 2/3 of triple-negative breast cancer have lost or show very low levels of PTPN12. Now, this is immunohistochemistry, so I think especially at a place like Yale, I think we could all argue that this is not quantitative by most measures. And I would argue that probably we're not seeing complete loss or inactivation at the protein level. Because when we look at pairs and matched PDXs, we can actually detect PTPN12 protein. But suffice it to say, if I can remind you that even a 50% change in dosage of this protein is actually enough to confer a phenotype. So we think that much of this loss of function that we're observing here is probably meaningful. Okay, so collectively, this data suggests that triple-negative breast cancers infrequently are compromised for PTPN12 function by mutation, and more frequently compromised at the level of PTPN12 protein dosage. And since I won't get into it later in this talk, I will tell you that we think that's through a post-transcriptional mechanism. Primarily in breast cancer, this is not a loss of the genomic locus or transcriptional silencing. Okay. Now, I referred to triple-negative breast cancer for the bulk of this talk as sort of the third subtype of breast cancer. But for those breast cancer aficionados, we all recognize that triple-negative breast cancer is really a grab bag of, it's hard to define a subtype of cancer as something that it's not. And so a lot of the molecular classifiers that have been overlaid on top of breast cancer have suggested that there may be 5, 7, maybe even 10 different subtypes of this subtype of breast cancer. And it draws into question really whether there are any common or universal features or pathogenic mechanisms in this breast cancer subtype. With more than 2/3 of this triple negative suffering loss of PTPN12, I would argue that that might be a prominent mechanism for this disease. And so with that, I think some of the obvious questions we began asking are, well, are triple negative breast cancers really dependent on that inactivation? We know that many cancers, when they inactivate or hyperactivate an oncogene or tumor suppressor, that they acquire a dependency on that. And certainly some of the elegant mouse models and human studies done right here in this room would attest to that, that behavior. And so we started asking very simple questions. Are PTPN12 deficient triple negative breast cancers dependent on that deficiency? And you can see here using retroviral transduction, restoring PTPN12 certainly has strong phenotypes in these triple negative models. If one does that across a panel of triple negative models, you can clearly see that a significant subset, but not all of these triple negative models, are very sensitive to restoration of PTPN12. And that suggests to us that this might be perhaps a general phenomena. That's in vitro. What about in vivo? To address that question, we've begun engineering many different triple negative models with inducible PTPN12 systems, again enabling us to ask treatment-like experiments. What happens when we restore PTPN12 function in engrafted tumors? And shown here is just one of our earlier experiments in which we've taken a metastatic derivative of triple negative breast cancer cells, engineered them with PTPN12 inducible cDNA, and you can see here induction of that cDNA leads to a prominent effect on primary tumor growth. We should also note that this elicits quite a strong effect on the lung metastatic burden in these animals, but interpreting that is certainly a bit confounded since one doesn't know if that's just an effect of the primary tumor borne out in the lung. And so to address that, actually with the help of Dawn, we began asking questions about what's the consequence of PTPN12 restoration in the metastatic setting. And shown here is just an experimental model of metastasis in which we have again induced PTPN12 function in the context of lung burden, and you can see a quite prominent effect on that metastatic burden. And I should say that this effect on bioluminescence has also been confirmed by histopathologic analysis. So collectively, this I think at least supports the hypothesis that triple negative breast cancers are dependent on PTPN12 dysfunction. And this led us to start asking some mechanistic questions about exactly why, how can we leverage this information from a basic perspective towards perhaps intervening in the therapeutic space. And so the obvious question is to ask, what are the substrates of PTPN12 that are important here? And for anyone in the phosphatase community, one realizes that's actually not a very, it's an easy question to ask, not an easy question to answer. And that is in large part, I think, been hindered by the lack of specificity of PTPs in vitro experiments. And so we began asking in a fairly systematic and unbiased way, what are those proteins that are candidate substrates of PTPN12? And we do this with orthogonal approaches, asking what proteins tyrosine phosphorylation is governed by this phosphatase, those that physically interact with and co-localize with PTPN12, and of course, genetic interactions always make us feel more comfortable. So using this type of approach, we think we've narrowed in on certainly a set of PTPN12 substrates, and I'm going to walk you through just a couple of representative experiments and then try to get to our translational data here in just a moment. So to begin asking, what are those proteins whose tyrosine phosphorylation is regulated by PTPN12? We went back to our engineered state in which we can inducibly deplete PTPN12 in mammary epithelial cells. So here we can study the tyrosine phosphoproteome in exactly two isogenic and paired states. And then using a modified version of SILAC, we can ask what are those proteins or peptides that become dephosphorylated or hyperphosphorylated when we deplete PTPN12. And we do this by simply labeling with heavy and light isotopes, mixing the peptides, enriching for those that are tyrosine phosphorylated, and then using LC-MS to quantify that change in phosphorylation. And using that type of approach, we identified a little less than 70 proteins whose tyrosine phosphorylation seemed to be strongly regulated by the presence or absence of PTPN12. And what was striking among those proteins is that there seemed to be a strong enrichment for actually receptor tyrosine kinases and their associated signaling, again, regulated by PTPN12. And our early data suggested that perhaps PTPN12 might be a negative modifier or a feedback inhibitor on these receptors, and that the lack of this receptor was then causing a pronounced amplification of these signaling cascades. And indeed, just a year or so after our initial publication, the late, great Tony Pawson published a really beautiful paper in Nature demonstrating, at least for EGF receptor, that this is indeed a feedback inhibitor that is recruited to the receptor within seconds to minutes after ligand stimulation. So this suggested to us a general model that PTPN12 might be a feedback inhibitor, an off switch, if you will, for some of these prominent proto-oncogenic receptor tyrosine kinases. And that's an attractive model, but of course is severely limited until one understands whether that's a general feature, all receptor tyrosine kinases, or whether that's something with a little bit more selectivity. And so we started narrowing our focus onto these receptor tyrosine kinases and asking of these receptors, which, if any, are bona fide substrates for PTPN12. And so we've been asking that question in a number of different ways. To do so, we cloned all receptor tyrosine kinases from the human genome. In this day and age of ordering your favorite cDNA from Genocopia or wherever, this seems like a pretty trivial thing, but it turns out in most of those cDNA collections, the receptors are either not sequence correct or not full length. So this was a bit of an arduous task. But we did clone out all receptor tyrosine kinases and then simply asked whether or not these receptors interact with PTPN12 by molecular fluorescence complementation, or biopsy, shown here, or by more standard methods, co-IP and others. And so there's really just 2 points that I want to stress here. First, PTPN12 does prominently interact with several of these receptor tyrosine kinases. But second, it's not a universal feature. So PTPN12 is not being recruited to all receptors, but it is a select set of these RTKs for which it might be a candidate feedback inhibitor or off switch. And so consistent with that idea, one can take triple-negative breast cancer cells and simply ask, what's the consequence of restoring PTPN12 function on signaling emanating from these receptors? You can see here several of these receptors that are PTPN12 interactors. Their signaling is significantly perturbed by the restoration of PTPN12. Some of these others, clearly no effect, and that certainly has an effect on downstream mediators like AKT, PI3 kinase signaling, and others. Okay, so that suggested by and large that this might be a feedback inhibitor onto receptor tyrosine kinase signaling. And I think combined with the functional data suggested that these receptors might be acting in concert or in redundancy to support triple-negative breast cancer. And consistent with that idea, we went back to the mutational data and made what I think is a fairly interesting observation. What I'm showing you here is a molecular replacement structure for PTPN12, the surface representation of that structure, where the colors are now indicating those amino acids with the most evolutionary importance. This is using an evolutionary action score innovated by our collaborator Olivier Lichtarge. And what you can see here is the hotter the color, the more evolutionary important are those residues in PTPN12. And what I hope you can appreciate is this pocket of red, which seems like a binding motif. And if one drills right into the protein, that leads to the catalytic cleft of PTPN12. What was intriguing is that many of the mutations that we were finding in triple-negative breast cancer and other cancers seem to be centering on this pocket, suggesting that they might be interfering with some molecular function or activity of PTPN12. And indeed, if one engineers mutations in PTPN12 and asks what is the consequence of those mutations on its molecular functions, you can see here that now they are partially, but not fully, hypomorphic for their ability to restrain, in this case, MET and PDGFR beta to what we feel very confident substrates of PTPN12. And a corollary to that, or associated with that, you can also see that these mutations are also loss of function for their ability to restrain triple-negative breast cancer growth, at least in vitro. So this is consistent, I think, with the idea that these tumor-derived mutations are defective in their ability to restrain these receptors and restrain triple-negative growth. And this and all the other data, I think, has led us to this, I think, fairly simple model. That PTPN12 suppresses breast cancer not by restraining any one given RTK, but instead by restraining multiple receptor tyrosine kinases in concert. And this model, I think, provokes some important questions. Mechanistically, we're still, I think, early days trying to understand how PTPN12 is recruited to and negates the function of these receptors. But I'd like to save that conversation perhaps for later in the Q&A. More broadly, I think from a translational perspective, I think this gives us some interesting leads or hypotheses. It suggests that in PTPN12 deficient disease, which I told you is quite frequent in triple negative, that these diseases may harbor hyperactivation of one or more of these PTPN12 regulated receptors. Now, I told you just a few slides back that actually these receptors, from a phenomena perspective, are already known to be hyperactivated in triple negative disease. And so we think this at least provokes the hypothesis that part of that hyperactivation in triple negative disease is stemming from a loss of this feedback inhibition. And so this obviously makes a translational prediction as well, that one might, in triple-negative breast cancer, rationalize the combination of receptor inhibitors towards these PTPN12-regulated kinases. And so we wanted to evaluate really whether that was possible, whether that mechanism or model holds true. And so, as you're aware, there are a host of different receptor tyrosine kinase inhibitors targeting this spectrum of RTKs. And so we began asking some very simple questions. In PTPN12 deficient disease, do these individual or combination of agents have any pronounced effects on cell survival? What I'm going to show you here in the interest of time is just some representative data, because I'd like to get to the translational work. Sufficient to say we did a lot of sort of combination matrix screens using not all receptor inhibitors, just those that target PTPN12 regulated kinases. And so we did a lot of these sort of simple two-way design dose matrix. One does a dose curve of one TKI targeting a PTPN12 receptor, another TKI targeting a different receptor, and you're asking for really the interaction between these two. And what I would say here is that we're not actually doing anything different from what I think pharma and other collaborators are doing in much grander scale. But because we've honed in on our molecular hypothesis, we can do it with quite a bit more depth and precision in looking at the cellular phenotypes. And so essentially what we're looking for are phenotypes like this, where a TKI targeting one PTPN12 receptor at a sublethal dose shifts the sensitivity of those of an independent TKI targeting another PTPN12 regulated receptor. And so to make a long story short, this is just a subset of the data, but you can see here what I'm plotting with this interaction effect. You can see here that the combination of Sunitinib and Crizotinib, which are inhibitors, of course, to PDGFR beta and MET, leads to quite a pronounced interaction effect in PTPN12 deficient breast cancers. The gray bars that you can't actually see here are those that are not deficient. And then if one looks at a broader stake of those models, you can see that PTPN12 responsiveness or the sensitivity to restoring PTPN12 tracks quite nicely with their interaction or their sensitivity to this combination. Now, Sunitinib and Crizotinib, we all know, have many targets, PDGFR beta and MET being just two of them. But I should say in data I'm not showing you, a lot of our genetic experiments, both loss of function and gain of function, suggest that PDGFR beta and MET are most likely the relevant, or at least two of the most prominent targets being affected here. So that's in vitro. What about in vivo? This was actually one of the earliest experiments we did. You can see here that in an aggressive model of triple-negative breast cancer, that the individual agents have little to no effect. That mimics what's actually been seen in the clinic. And you can see here that the combination certainly has an effect that looks more than additive. So this was very encouraging from a sort of mechanistic perspective. Perhaps this might give us some therapeutic traction. But actually, I think it also gave us reason to pause. And that was for two reasons. First, I told you, first I would say is that for any therapeutic response, one is looking for more than stabilization of disease. But more broadly, I just finished telling you triple negative is probably this grab bag of many different diseases. And so I think the question is whether this therapeutic entry point has any traction across that heterogeneity. And so for that, we turn to our long-standing collaborator, Mike Lewis, who at Baylor has been establishing a bank of patient-derived xenografts, or PDX models. I know that there's many different efforts here at Yale and elsewhere to establish those for different types of malignancies. We've been focusing a lot on breast cancer. And Mike has, I think now, at about 40 different chemorefractory models that came from triple-negative patients. And he, of course, has been profiling these on multiple different platforms. But for us, what this gave us, in addition to a wonderful repository for discovery, is a forum to test this hypothesis that perhaps PTPN12-deficient triple-negative breast cancers are sensitive to Sunitinib and Crizotinib. And so for that, we, of course, first started by assessing PTPN12 deficiency. And you do indeed see that deficiency in the PDXs mirroring the primary patient population. Actually, the very first model that we tested was a PTPN12 deficient model. And you can see here, and this model is certainly sensitive to the Crizotinib+Sunitinib. This is a pronounced and durable response, although I will tell you, we have not done the experiment where we take animals off of drug and measure that durability off treatment. So this suggests, in one model at least, that PTPN12 deficient triple-negative breast cancers are very sensitive. What happens if you take a snapshot across these models? And that's shown here. So what I've done now is summarize the effect of each independent PDX, their response to Crizotinib+Sunitinib. And so each individual symbol here is actually an independent PDX represented by many mice. I tried to denote that here, where I'm blowing up from this one PDX model. You can see this is upon vehicle progression. You can see pronounced effects in the Crizotinib+Sunitinib population. And I think it's pretty exciting on a couple of fronts. First, we actually noticed regression in disease in roughly half of the animals, or a little more, in the context of Sunitinib-Crizotinib combination. I should remark that the way I've plotted it here is a little bit of underestimation. We're not getting the max effect here. It's just at a static time point when the vehicles have progressed. But I think the pattern, or the trend you can see here, is also that PTPN12 deficient disease also tends to be very responsive to Sunitinib and Crizotinib. And if one looks at a simple survival or progression-free survival, you can see that while there definitely is an effect on these molecules on both classes of PDX models, certainly that effect is more pronounced in PTPN12 deficient disease. So with this data at hand, that was the impetus, really. We've started now a clinical trial in collaboration with Pfizer, asking the question, are patients with PTPN12 deficient disease responsive to this combination? We have a phase 1b design that's going on now, and hopefully move to phase 2 quite quickly. More broadly, I think that this raises an interesting possibility for the use of receptor inhibitors that are, of course, quite common on the shelves of our pharma collaborators. Certainly, those inhibitors have been utilized and leveraged in the context of receptor mutation, where that ascribes both the target and the responsive patient population. But we think there might be even a broader swath of patients that have suffered loss of feedback inhibitors like PTPN12, and that if one had better maps of PTP receptor antagonism, this might lead us to some new ways to think about employing those inhibitors in the clinic. And so with that, the most important slide is certainly to thank those who've contributed to the work. This is actually a partial list. We've benefited tremendously from wonderful collaborators at Baylor and abroad. And certainly I want to draw your attention to Tiffany Sue and Amritha Nair, who were the students who led both of those two projects I just showed you. If you're looking for postdocs, you'd be lucky to recruit them. And with that, I thank you for your attention and be happy to take any questions.